Our next presentation is from Dr. Alexander Varshavsky, who is the Smits Professor of Cell Biology at the California Institute of Technology. He received his Bachelor of Science degree from Moscow State University and his PhD from the Moscow Institute of Molecular Biology. He spent 15 years in the Department of Biology at MIT and then joined the Division of Biology and Biological Engineering at Caltech in 1992. Dr. Varshavsky was awarded the Breakthrough Prize in Life Sciences last year for discovering critical molecular determinants and biological functions of intracellular protein degradation. He's an expert on the Ubiquitin system, a ubiquitous regulatory protein found in all living tissue and now known to be involved in nearly every aspect of cell biology. Alexander.
Thank you very much. As everyone who received Breakthrough Prize, I'm of course very grateful to Yuri Milner and his colleagues who thought about it and did it. I was asked by Yuri in particular to talk less about what I do for a living and more about the future, which I don't have the slightest idea about. And the only safe thing about predictions is they tend to be off by a mile. And so the question is what to do, given 20 minutes that I have. What we will do is the following. I will try to show you the slides that we might return to if the future will take short time to describe. And if it takes longer than I think, we might simply assume that everyone knows about Ubiquitin system. It is shown here. It's a small protein. It gets activated through enzymatic cascade. All of that has been figured out over many years by many people. It eventually is joined as a polyubiquitin chain, which we discovered many years ago, to a substrate, that yellow thing, and that yellow thing can be just about any protein in the cell, provided it carries a degradation signal, some feature, which we call a degron. That could be a lot of different things, and our laboratory many years ago also discovered the very first degradation signals, the primary degradation signals that actually suggest the ubiquitin system to target the protein. And then such a contraption, a substrate to which polyubiquitin chain is joined, is destroyed processively by a complicated multi-subunit ATP-dependent device called 26S proteasome, and I'm not even remotely doing justice to the complexity of this system. The real complexity of the system resides in this seemingly simple thing called E3, part of the ubiquitin ligase. About 1,000 of distinct E3 ubiquitin ligases are encoded by our genome. It's a larger number of different ligases than the number of phosphokinases, for example. The immense functional reach of the ubiquitin system, it does just about everything in the cell, comes entirely from this immense multiplicity of ubiquitin ligases, which are specific in general for different degradation signals, different degrons. And that's where the reach of the ubiquitin system is coming from. This is the ubiquitin fusion technique invented in our lab by Andreas Beckmeyer, Dan Finley, and myself 28 years ago. It led directly to the discovery of the very first degradation signals at that time in 1986, and the discovery of the pathway, part of the ubiquitin system, that underlies these signals, which we call endegrons. And they're called endegrons because they're startlingly, or at least seemingly, simple. They involve a single N-terminal residue of the protein. A single N-terminal residue of the protein in the presence of some kind of right configuration interiorly, which I will skip because we don't have time for it, is sufficient to regulate the protein half-life. And the half-life range from very highly stable, many hours, to less than a minute, about 10,000-fold range, is produced essentially by variation in a single residue at the end of the protein. And that, of course, is still the smallest and probably best characterized degradation signal there is. the underlying pathway is, of course, quite complex and took our laboratory a very long time to decipher and to begin to understand. In fact, 4 years ago, we discovered another NN rule pathway, because it turns out that in addition to the first NN rule pathway, which we now call the arginine rule pathway, there is a second NN rule pathway, which we now call AC NN rule pathway. The reason for ARG and AC comes from two specific features of the NN rule pathway. Since we don't have time to go into all these gory details, I will tell you very simply what it means. If a protein emerges from the ribosome, the first thing that emerges is the N-terminus. The N-terminal residue lights up targeting by two NN rule pathways, which are present universally in all eukaryotes. Bacteria also have an NN rule pathway, but that's a separate discussion. These two NN rule pathways see the N-terminus in two entirely different ways. Arginine rule pathway attacks unacetylated N-terminal residue, whose alpha amino group is free. AC NN rule pathway, discovered by Chel-Sang Huang, Anna Shimori in the lab only 4 years ago, in 2010, attacks N-terminally acetylated N-terminal residue. It is N-terminally acetylated because N-terminal acetylases hug the ribosome. They are weakly associated with ribosomes, and they co-translationally anti-acetylate approximately 90% of human proteins that emerge from the ribosome. And the moment they emerge, they are attacked, unless they find an abode, a cave, to hide their end. So this system actually targets the first checkpoint that a protein ever encounters are these two NN rule pathways, the view that emerged relatively recently. These two NN rule pathways attack newly emerged nascent proteins from two sides. If they're unacetylated, if they somehow skipped being anti-acetylated, arginine rule pathway attacks them. If they're anti-acetylated, AC NN rule pathway attacks them. And the only way to escape that attack is to become a part of multi-subunit complex, or to collapse into a properly folded structure in which the N-terminus is hidden in the armpit of the fold, or to escape topologically through translocation across the membrane into the, for example, lumen of ER or matrix of mitochondria. And these are the fates of nascent proteins. They're immediately attacked by these two NN rule pathways unless there are stochastic escapes into folding, assembly, or translocation. And so those two pathways, this is what we are seeing is the yeast NN rule pathway. And these are groans, for which we don't have time. This is mammalian NN rule pathway, the arginine rule pathway. And as you see, it's slightly more elaborate than the Cerevisia NN rule pathway, but it has all these features. For example, and I will mention just one feature because it will be important in discussing the future possible developments. If, for example, a protein has aspartate or glutamate at the N-terminus, it is not recognized directly by ubiquitin ligases of the NN rule pathway, which are over here, which we call N-recognize. It has to be first conjugated to arginine, becoming one amino acid longer before it is destroyed. And the enzyme that conjugates arginine is called arginyl transferase. We're working on this enzyme, and it took a long time to decipher this actual circuitry. And you will see why I mentioned this fact a little later. These are all kinds of functions of the NN rule pathway. Both of them have a number of different functions, in fact, striking number of different functions. The arginine rule pathway has been studied by now for 28 years in our lab and later by other laboratories, some of them by the former postdocs in the lab. And the ACNN rule pathway is just a child. It's only 4 years old, and it's already becoming a field. This slide I hope to return to if we do not get bogged down by the future. And this is future. I wanted to tell you about two ideas, childishly simple. I would not even dignify them by calling them ideas. Okay, they're so simple, as you will see. I had them both about 25 years ago, and I we never found the time to develop them in the lab, and I was absolutely I was certain that somebody will pick it up and develop it totally independently, because I couldn't believe that these ideas cannot occur to anybody else but myself, and I'm pretty sure that they probably occurred to other people, but I never saw them published. And they have never been implemented still. They're still ostensibly new ideas, although, of course, they're not new, and they're so embarrassingly simple that, again, I don't think they're really ideas. Here they are. Here is the first one. You want to have some control over the level of active protein in a cell. In fact, many inhibitors, many drugs of today are nothing but just simply down-regulators of a function of a specific protein, if they're good drugs. Well, there are many ways to do that. One is by taking a small molecule and asking it to bind to a protein and inhibit its enzymatic activity. Well, given what we're discussing, there is another method to do it, to make a protein short-lived, to destroy it. It will be equivalent to inhibiting it. Well, how to do it? There are several, in fact, there are many different ways to do it, and I will skip ways that are already being pursued in a variety of different ways. For example, making a ubiquitin ligase to recognize this protein and to build a polyubiquitin chain and to target it to the proteasome. This way of doing things is already being implemented, and there are some interesting results, and indeed, I think it will work eventually as an approach to drug development. It already is working, I think. But what I suggest is a slightly different approach to the same end. As you already heard, if you have a destabilizing residue at the N-terminus of the protein, the N-and-rule pathway may come in if other conditions are met, and most of the time those other conditions are met, and destroy the protein by targeting it via the proteasome and processively degrading it. How to do it? Well, imagine that you develop a restriction protease. There are already some restriction proteases. Many of them are viral proteases that recognize a specific region in a polyprotein of a virus, for example, and cleave. So all you have to do is to take a protein of interest, to find some kind of a loop in this protein, which has unique sequence, and develop a protease that cleaves this protein just a single time. Okay? The protein does not necessarily fall apart because it will be folded, and cleavage may not by itself inactivate the protein, but make sure, and this is the idea, it's very simple, it's not really an idea, it's so simple. Make sure that when you cleave it, the C-terminal part of the cleavage site is a destabilizing N-terminal residue. Then the N-and-rule pathway, being a mechanochemical device, comes in, dissociates the protein, and destroys the C-terminal part of the protein. And if you place the cut sufficiently close to the N-terminus of the protein, it will destroy the bulk of the protein. So that's the first idea. And there are all kinds of interesting collaborations for which we don't have time. Believe it or not, it has not been done. And if such a small protease, a restriction protease, can be developed and can become cell permeable, and as many of you know, cells can be penetrated by fairly large macromolecules if they're designed right, and this kind of art is continuing to improve and will become possible better and better in the future. This is not necessarily such a far-off approach, and I just hope that either us or someone else will pick it up and develop it into a real approach. The second approach is actually slightly different, and that is if we return now to arginyl transferase. Notice what arginyl transferase does. It conjugates free amino acid, arginine, via arginyl tRNA, to the N-terminus of the protein that has either glutamate or aspartate or modified cysteine residue, I will skip that, at the N-terminus. Well, generalize the function of this enzyme. Make this amino acyl transferase recognizing not a single residue at the end of the protein, but a sequence, a specific unique sequence at the end of the protein, and then join something like arginine, to the end of the protein. What will happen? It will become an N-rule substrate and will be destroyed. So the other second idea that is also not implemented anywhere by perhaps directed evolution of arginyl transferase, I think it's entirely feasible, is to create an amino acyl transferase that recognizes the N-terminus of any specific protein simply because of unique sequence at the N-terminus and conjugates a destabilizing residue to that protein and thereby makes it a substrate of the N-rule pathway. Is it feasible? On paper, for sure. Realistically, I also think so. Is it done? Not done. And so I think in the next decade, we actually, I repeatedly tried to convince a couple of people in the lab to try to do it. I was so far unsuccessful, but I don't give up. And on top of that, I'm sure that other people might be interested in such simple approaches and eventually implement them. So these are two simple examples of how rich is potential future of advances when even such very simple ideas have not been yet realized and implemented. And before, since I want to tell you a little bit about how I see the remote future of medicine. I don't see it in any original way at all, but I wanted to describe to you an analogy of sorts. That's how I see our current times, that we're embedded in our times, and we look backward, but we cannot quite look forward. I think after discussing all these short-term advances, it might be interesting to discuss a longer-term view of the future of medicine. I see this future as being realistic thermodynamically, so to speak, i.e. it should arrive one day, as there is nothing in the laws of physics that precludes it from arriving, but uncertain kinetically, i.e. we cannot be sure when actually such a future will arrive. I think the times we're living through are likely to be analogous in the sense that I will try to define in a second, to roughly 1830s, when Michael Faraday was discovering in London the effect of electromagnetic induction and was making first tiny devices, still laboratory curiosities that we would call today the first electric motors. The discovery of electromagnetic induction, which is of course a change in magnetic field that induces a current in a nearby conductor, or a change in the current that creates a magnetic field that can act on a nearby piece of iron, that's what electromagnetic induction is, led to two profound consequences. First, it resulted in the James Clerk Maxwell's theory of electromagnetism. That theory was based in part on the Faraday's experimental discovery. Maxwell's insights led to a major revolution in our understanding of the world that eventually gave us the theory of relativity in quantum mechanics. Second, and I find it really remarkable, the same Faraday's discovery has led over next several decades to the appearance and rapid evolution of electric devices of all kinds, including generators, electric motors, and later, much later, modern electronics. Faraday would be shocked to see what his discovery has become today, both in modern physics and in everyday life. I think that the enormous and exhilarating progress in fundamental molecular biology and related areas today is at a deeper, well, much deeper level, is akin to rapid advances in fundamental but not yet applied physics at the time of Faraday. The enormous long-term consequences of discoveries of the kind that Faraday was making at that time, during that time, could not be glimpsed by even keenest eyes. The discoveries today in fundamental molecular biology have already begun to influence the practice of medicine, but major diseases, let alone aging itself, are still with us. Future applications of the molecular level understanding of biology that is being attained now would be enormous and would stand on the shoulders of current discoveries. Some of these discoveries may seem esoteric, but would be anything but a generation or two from now. In other words, the ongoing discoveries and inventions would soon lead to a veritable, as physicists would call it, phase transition, a self-accelerating transformation of medicine into a discipline of immense power an amazing subtlety that is next to impossible to glimpse from where we are today. Hence, this analogy that I'm suggesting to the times of Faraday. The fundamental genetic, biochemical, cell biological, and computational understanding of living cells and multicellular organisms would give rise within 1 to 3 generations, closer to 2 generations to 3, I think, to major revolutions in medicine that would be based on basic research that the Breakthrough Prizes are given for today. These revolutions would make human diseases, literally most of them, a solved problem. As a result, physical accidents would become by far the main cause of death. Aging itself may also be tackled decisively based on its thorough understanding, in that the human aging would be slowed down in a major way. Humans are already becoming partly quote-unquote bionic, with complex prosthetic devices being routine already. And this trend would expand during coming decades and may also involve direct manipulation of the human germline, perhaps through developments of CRISPR-like technologies that are becoming better by the hour. Biologically informed, technically sophisticated alterations of DNA in the human germline would be a long time in coming for all kinds of reasons, including ethical considerations, but would be essentially inevitable once the means for doing such alterations become mature enough. As most of us know, once answered prayers are a dangerous thing. Will the utterly new world that I try to depict be a better place for happiness, serenity, and other good things? I know not, but hope it will be. And this is an attempt to predict a remote future. What I will do in the next literally, I don't know how much time I live, literally 3 minutes maybe, is to focus on this slide that basically shows you what these two NN rule pathways can do together. They are fundamentally complementary processive proteolytic systems that have in common a topological property. They recognize the N-terminal residue of a protein. Arginine rule pathway recognizes unacetylated N-terminal residue. And AC NN rule pathway recognizes acetylated N-terminal residue. One last thing to know to understand slide B in this larger slide is to know that when you anti-acetylate protein, because it evolved to be anti-acetylated under normal conditions, it will actually bind within its cognate complex tighter, about 10 to 100-fold tighter, significantly tighter, than it would otherwise be if it doesn't have this extra N-acetyl group. Because if it doesn't, it creates a cavity, and there is a thermodynamic penalty for a cavity. What the result would be, it turns out, and this is an experimental fact, that many, perhaps most, human proteins, when they're anti-acetylated, are anti-acetylated only partially. 90%, 95%, sometimes 70%, because the event is stochastic. At the exit from the tunnel, this train is hurtling past you, and if you're anti-acetylase and missed your chance, you cannot do it again effectively. So the result is that there are many not anti-acetylated proteins swimming around in the cell. It turns out, and there is already evidence for that produced recently by Anna Shemori and Cheryl Sang Huang in our lab, that if the protein is not anti-acetylated and becomes a part of its cognate complex, because affinity penalty is about 10 to 100-fold, it keeps dissociating. It keeps dissociating from the complex. And when it dissociates, it's pounced upon by the arginine rule pathway. So the result is a protein repair. Repair of protein complexes by virtue of the fact that the absence of anti-acetyl group makes this subunit an unstable member of the assembly. And that subunit dissociates, and during its dissociation tends to be destroyed, so that it can be replaced by anti-acetylated subunit. So the result of this complementarity of the two NN rule pathways that attack newly formed proteins when they're formed is that not only checkpoint of quality control is possible, because misfolded proteins, of course, will not fight find a safe abode easily and therefore would be preferentially destroyed, but also protein remodeling because non-anti-acetylated protein that actually found abode discovers that that abode is unsafe because it doesn't stick there for very long. It dissociates, and during dissociation gets destroyed, and anti-acetylated protein takes its place. So there is a great deal of beauty and function to these N and rule pathways, and I will stop now. Thank you.